Biologists often use optical microscopes because they are a simple way to image biological samples without damaging them. However, all optical microscopy is restricted by the diffraction limit, which says that if two fluorophores are too close, they will appear as one fluorophore under a microscope. We use the word resolution to describe the ability to distinguish two objects that are close together. So what the diffraction limit means for optical microscopy is that our resolution is always limited to a few hundred nanometers. To get around the diffraction limit, scientists have developed many different super resolution microscopy techniques. However, these super resolution techniques have their own downsides. They're expensive, they have longer imaging times, and require more expertise. Expansion microscopy is a super resolution technique first introduced by the Boyden lab in 2015 that circumvents the diffraction limit by physically separating fluorophores to greater distances. The way it works is by embedding the sample within a polymer gel, which expands when you add water. The sample is linked to the polymer gel, so when the gel expands, so does the sample. The sample expands isotropically, which means the shape is preserved. Using this technique, we can achieve higher resolutions. For example, if we expand the specimen by 10 times, we can achieve a 10 times greater resolution than we could before. Expansion microscopy is performed in 4 steps. The first step is anchoring. In the anchoring step, we want to give the sample away to attach to the gel. The most common way to do this is to introduce a chemical called ACX, which can bind to the amine groups of proteins, including the amine groups of fluorophore proteins. The second step is polymerization. In the polymerization step, we infuse the sample with sodium acrylate and a crosslinker. The crosslinker helps sodium acrylate form polymer chains and also forms crosslinks between those chains to form a polymer network. The ACX anchors have an acrylate group, which also bonds to the polymer network. The third step is homogenization. We need to make sure the sample expands evenly when the polymer gel swells. In other words, we need to make it mechanically homogeneous. This is done by treating the sample with protease, which is an enzyme that breaks down all the protein within the sample. Luckily, most fluorophores are protease resistant, and enough linkages between the fluorophores and the network have formed so that even though the sample is destroyed, we are left with a fluorophore phantom that appears the same to a fluorescent microscope. The last step is expansion, and this is done by adding deionized water. After multiple washings, the network can expand between 4 and 10 times its size. We can also polymerize a second gel and expand the sample again to achieve an even higher resolution. Because of its speed and ease of use, expansion microscopy has many applications, such as in biological research, where it has been used to image cells, tissues, and even entire organisms. Researchers have also used expansion microscopy in a clinical setting where it can act as a cheaper, faster replacement for electron microscopy. In one study, expansion microscopy was used to successfully diagnose kidney minimal change disease. Another study used expansion microscopy to create high-resolution images of breast tissue, which were used to train a machine learning algorithm for classifying normal, benign, and pre-invasive malignant tissue. Expansion microscopy breaks the diffraction limit in a rather straightforward way. We can create a fluorescent phantom of whatever we are interested in and expand this so that previously diffraction-limited spots can be resolved. It has its limitations, the biggest one being it is incapable of imaging living cells, but its main strengths are very strong. It gives biologists a cheap and fast way to produce super resolution images, and it is compatible with many existing sample preparation methods. In the future, different variants and optimizations, as well as the possibility for higher resolutions, make expansion microscopy a very promising technique.